All right. Good afternoon, CGIL. This week, we are happy to have Dr. Tad Sonstegard, the Chief Executive Officer at Excelligen, presenting for us. Dr. Sonstegard oversees the commercialization of groundbreaking genetic improvement breeding platforms for food animals that combines genomics, gene editing, and advanced cell culture and reproductive methods. Under Dr. Sonstegard's leadership, Excelligen has moved from making proof-of-concept animals to achieving the first commercial use approvals in multiple countries with their innovative product lines for heat-tolerant beef and dairy cattle. Prior to his role at Excelligen, Dr. Sonstegard directed a distinguished livestock molecular genetics research program at the USDA in Beltsville, Maryland. His team pioneered the first commercial agricultural-based SNP tool in 2007, enabling genomic selection in cattle breeding. Recognized with multiple tech transfer awards, this work transformed genetic improvement for progressive animal breeders and genetic companies. His academic journey began at Iowa State University for his undergraduate degree, followed by a PhD from the University of Minnesota after leaving his family's Red Angus cattle operation in west central Minnesota. Today, Dr. Sonstegard's presentation is on the adaptive breeding of performance cattle breeds. So I will turn it over to you now, Dr. Sonstegard.
Thank you, Gabriella, and thank you for the invitation to speak to CGIL. What I'd like to do today is just take you through a bit of what's been going on the commercialization side for gene editing technology, and talk a lot about what we're trying to do, especially to affect genetic improvement in the tropics. Just a little bit about us, some background. Of course, we have our mission and vision statement there on the left, but we're really a company that's empowering partnerships with progressive breeders to bring this technology as a breeding tool for rapid genetic improvement in a single generation. But the whole concept of gene editing in livestock cells was initiated when 5 professors from the University of Minnesota formed the parent company Recombinetics. And there's really multiple applications for these gene-edited livestock cell products that include not only genetic improvement of food animals through precision breeding, but also applications related to improving biomedical knowledge through testing of human genetic disease models in swine, device testing, and also livestock cell products that can be used to increase human longevity through xenotransplantation. Currently, we have only 18 employees. We're very lean. And of course, the Excelligen part is dedicated solely to gene editing on food animal cells that can be turned into animals that can be used by breeders for genetic improvement. The original parent company was formed in 2008, but Excelligen was formed as a marketing arm in 2013 and really wasn't operational until I joined in 2015. Most of the capital that's been used to develop the products, not only on the biomedical side, but also the food animal side, has come through angel investors and non-dilutive capital, which is grants. We did have a Series A investor back in 2019, and I can tell you that this technology is very expensive to develop and to bring to market. Part of that expense, of course, is you have to hold licenses in order to use the technology to make commercial products. You also have to file patents to protect certain product lines that would include not only the methods for delivering edits to cells, but also some of the traits that we employ. And we've developed proof of concept traits in 3 different food animal species, so both beef and dairy cattle, swine, and finfish. This is just a timeline with some examples of the types of animals that we've made over the past 10 years. You can see in the early days, it was just about testing different gene editors and proving out whether or not we could introduce those traits through breaking DNA and directing the cellular repair to recode the DNA to express the trait that we want. And then around 2019, we started using CRISPR-Cas9 instead of TALENs, and we started accessing actual elite commercial genetics and putting the traits into that rather than just using experimental lines of animals that we could get from universities. And so we do have commercial partners. We have a number of projects going on where we're developing commercial lines with partners or solely on funding internally through Excelligen. And so this just represents all of those, and I'll go through some of these during my talk. But you can see that we have a very broad experience, and we've made hundreds of different animals, founder animals, through gene editing. So we are the pioneer company that offers this technology to outside animal breeders. Currently, there's probably only one other commercial entity that's used gene editing, and they've tried to put a disease resistance trait into pig. They still are not commercialized. Currently, we have 3 different traits that have been approved as non-GMO in multiple geographies, and also we have a limited enforcement discretion in the US, which I'll talk about later. And then we have 5 other traits that are still pre-commercial, which means we still have to do some R&D in order to evaluate their economic value going forward. We have achieved non-GMO commercial allowances, like I said, on heavy muscle polled and slick beef and dairy cattle in 4 different countries now. So that includes Argentina, Colombia, Brazil, and the US. And we've sold semen in the US, and I see one of the attendees of this talk has bought some semen for use in Brazil. So that's what the Brazil Canada represents. And also we have progeny on the ground in the US in multiple locations. So really, during my time with the company, which started in 2015 after I left USDA, our focus has been not only on finding better ways to introduce edits into our germline cells that we can breed animals from, but also on how do we monetize gene editing technology as part of the continuum of breeding tools. So if you go through this continuum, I know in the developed world, we have access to all of these technologies. And some of these technologies you actually need in order to use the gene editing technology appropriately in the commercial space. So that would include the reproductive technologies because you have to edit the cell that can be turned into an animal. And then, of course, you want to use the type of genetics that's already within a genomic EPD program. So you're actually editing the best of the best because you want these animals to be used extensively. So they need to really be at the top of the breeding pyramid. In many places where gene editing technology can have the biggest impact, of course, some of these technologies still don't exist or they're very rudimentary. So making the use of gene editing in those low middle income countries or emerging economies is much more difficult. So why would you use gene editing for animal improvement? So there's some very obvious choices to introduce traits that we're missing in our breeds. So we know most of the dairy animals are horned, and animal welfare groups find dehorning, the process of it, somewhat distasteful. So do the farmers. So adding polled into horned breeds that are already very elite as far as their protein production performance, that would be one example. And heat tolerance and temperate breeds, which I'll talk about more extensively. A second application would be to eliminate emerging genetic disease. So I give an example here of one of the blood disorders in Wagyu cattle because of the close population in the United States. There's not a lot of diversity, and many of the animals are carriers for some of the different blood disorders. And then I give another example of bovine congestive heart failure, which is really an emerging genetic disease in Angus where animals lack oxygenation as they get very close to slaughter weight and keel over and die in the feedlot. Probably the biggest promise of this technology, though, is really to protect our animals from pathogen-induced infections. We know it's very costly to challenge our elite germplasm with disease challenges, and a lot of times the work we do in vitro doesn't translate over into genetic solutions for our breeding. And vaccines are not always effective. So here I just list some of the diseases that we actually work on as far as trying to provide better resistance or resilience in our animals to these diseases. Certainly, many of them are, at the root of them are polygenic effects within the genome as to whether or not an animal is susceptible or resilient. And then the last category I call specialty products. You may have seen in the media what I call avatars or surrogate sires, where you ablate the germline cells through knocking out a gene so that it doesn't express the ability to produce sperm in the testicles. And then you would transplant spermatogonia from another bull or a different breed into that avatar animal to have it produce semen from the donor. Other things would be having bulls that only have offspring of a single sex, or have offspring that when they're born are sterile so they don't have to go through surgical castration, or it's a way to protect your germplasm from being taken by a competitor, which is somewhat typical possibly in the aquaculture industry. If we think about the spectrum of the difficulty for some of these edits, really, if you're talking about monogenic traits, it's simple. Typically, we're talking about naturally occurring alleles, and in most countries that follow the Cartagena Protocol, these would be considered non-GMO. And SDN really stands for site-directed nuclease, so that's just using any sort of one of these enzymes that you can target to break the DNA in a specific place, and SDN1 is what would be used if you're just targeting a region of the genome and allowing the cell to repair it. SDN2 would be what is called homology-directed repair, where you actually provide some sort of a splint, typically in the form of an oligo within our company, to bring that double-strand break back together, but to recode it the way that you want. And then SDN3 would really be adding a foreign gene or a transgene. And so along that spectrum, there's much more complex editing that could be multiplex editing, or using what we call rational design, where you know a gene that's involved, but there's no natural variant for it. So you remove some motif or alter the expression of that gene to express your trait. And then, of course, GMOs when you add a transgene. So what have we learned from using these applications and some of the traits we've been doing in our proof of concept animals over the past decade? There's still very few major gene editing targets of economic importance, but I would stress that the ones that do exist have still yet to be fully commercialized. And I think for us, our lead trait is still heat tolerance in cattle. This is just some data from back when I was at USDA and was collaborating with the University of Puerto Rico in Mayaguez. And there are many, many cattle there that have the what we call SLIC1 version of the prolactin receptor. And then they have Holsteins that also do not contain that. Most of these Holsteins have been bred up from native cattle, where that trait was very prevalent. And when we segregate those based on genotype and then look at their production or fertility differences, we can see that in a tropical grazing dairy like those typical in Puerto Rico, the SLIC trait provides a performance advantage to those Holsteins as far as being able to produce milk and be rebred in a tropical zone. So we thought that this is a pretty good economic value and a way to improve dairies in the tropics. We did a lot of studies on Criollo cattle along the way. I have a student out of Colombia that's just graduated with her PhD from Brazil, and he did a lot of surveying of different Criollo breeds in the Caribbean basin. And we found 6 different versions of the SLIC mutation in the prolactin receptor, all of which cause this phenotype of short hair that allows these animals to cool on the exterior, but they also regulate their internal body temperature at least a degree Celsius lower than a normal conventional animal. So they're very much buffered to withstand that tropical heat. So with that in mind, instead of doing what's been done by some of the breeding companies, and you can see SLIC Holsteins available that have been created through admixture breeding, which I show here on the left, which just involves a lot of back crossing to bring that genomic merit back up after you introduce that SLIC trait. You can just do this in a single generation with any of your elite germplasm using CRISPR-Cas9. So the issue with the conventional breeding, of course, it's very costly in time and resources, and it is disruptive to genomic selection. Whereas within the gene editing, we're just mimicking the natural mutation by breaking that DNA and telling it how to repair. And we can put all of this under a quality control process, and it's not going to be disruptive to a genomic selection program. So we can identify the most elite animals and use those either through embryo editing or cell editing those cells are clonable, and clone those animals to provide those slick dairy animals in a single generation to be used in the tropics. If we look around, there's of course been many academic studies, but very few companies in this space. But many of what you see, not only on the animal side, but in the crop side, is breaking genes is really the simplest use of this tool. And that's great for R&D models, but it's not always the best for commercial use, especially if you're thinking about introducing gene edits into embryos, and I'll talk a little bit about that. But here's a project where we were just breaking what is the kisspeptin gene to create what we called our castration-free swine. And here's just showing a results table for both the male and female side. And what we're able to show is that the animals that were not edited, that were produced from embryos not edited, they of course had normal development, body weight, gonadotropin levels, and they also produced bordetine. So part of why we're doing this genetic castration is to lower bordetine, because we know that that's the main reason why swine are castrated, is to get rid of the distasteful flavor of pork. And if you look at the phenotypes, you can see that when we knocked kisspeptin out fully, and we did this, we had to breed these animals out to the next generation to make sure that we only had 2 alleles, because when you inject embryos with CRISPR-Cas9, sometimes not all of the cells that are turning into that embryo get edited, so you end up with what's called mosaicism. But you can clearly see when kisspeptin's knocked out, these animals don't enter puberty. Their gonadotropin levels are very low. Their body weight is the same, so they grow very similar. And they don't produce any bordetine. The heterozygotes act pretty much like the wild type. And so if you want to learn more about this project, of course, it's detailed in this publication I show below the table. The problem with this trait is that once you make an animal sterile, how do you keep that genetic line for breeding to be able to propagate this trait to its offspring in future generations while still doing genetic improvement? So in order for this trait to really work in a commercial setting, you have to do fertility rescue, which is an ongoing project that we're collaborating with the University of Nebraska-Lincoln and also US Mark at Clay Center, Nebraska. Next step really is what types of cells can we do editing in and really come up with standardized breeding outcomes. So in that last slide, I showed you what the result of injecting embryos is. You end up with mosaic animals, and you have to do some breeding in order to get the full expression of the trait. The issue with gene editing in embryos is really inefficient to make exact changes to genes. So a lot of the gene editing targets we have are just making changes to the amino acids. So you would just recode the protein that's being expressed, or you're doing some sort of a truncation of that protein, but you want to have the exact same thing that exists in nature. So proof of that is we did a very large IVF embryo injection project to create a slick line of Angus cattle. So we owned a number of Red Angus donors. We purchased semen on the commercial market. We created those IVF embryos, and then we added our CRISPR-Cas9. And the idea was we would add the HDR oligo to some, and in some we wouldn't. But our most efficient use of these embryos was really just doing the NHEJ or the SDN1. And that qualifies in a number of geographies because you're not using a template as non-GMO. So we're also thinking about the market ahead of time. And we made some of the first black-red carrier animals actually that are slick. Here I'm showing one of the females, and she was the first registered gene-edited animal in the United States. And she was registered through the Red Angus Association in the United States because we have two different breed associations for the coat color in Angus. Here's that SO3 again next to her full sib sister SO2, and this picture was taken in June. You can clearly see at this yearling age that there's a tenfold difference, which we measured through shaving the hair in their coat. So the animal that did not get the edit, of course, she's expressing the normal amount of hair as a typical Angus animal, whereas the crisper cool girl on the left there is clearly slick. So easily observable phenotype. But does it actually have any effects on their ability to regulate thermal tolerance, growth, and reproduction? And so we were funded by FFAR, which is a USDA agency, and we collaborated with CMEX and the University of Florida to make both Angus and Jersey animals using this embryo approach. And what we found was in these founder animals, they had a superior ability to regulate body temperature in a Florida environment. The gene-edited animals displayed superior growth and product yield, and there was no deleterious effects on reproductive function in either sex. So that a lot of times you can see opponents of this technology worrying about, well, when you're affecting prolactin, you may be affecting lactation or reproduction. And clearly that's not the case, especially since we're mimicking naturally occurring mutations that have evolved because they give an animal advantage in a hot environment. So if you want to read more about this, of course, the paper is listed here from Facet Bioadvances. Okay, so that's probably the simplest example and still a trait that needs to be fully commercialized. And there's a lot of potential in tropical beef and dairy markets for its use. But really, if you look ahead, stacking edits is going to be the future. And what does that mean? That's just editing multiple genes to exact changes and doing that using homology-directed repair. So there are crop varieties that can make through gene editing. A lot of times those are just knockouts. Doing knockouts is pretty simple, but doing HDR at multiple locations is much, much more technically difficult. And why do we want to do that? Because we know we've already been able to do single genes in creating our polled animals or heat tolerant animals, and also knocking out myostatin to make increased muscle. And this is just, again, showing CRISPR cool at an older age. But what happens if we have the ability to do multiple genetic changes is we can really begin to address improvement for disease resilience, enhanced efficiency, and possibly carcass quality traits that would include improving marbling, let's say, in zebu lines of cattle from the tropics, as an example, or milk production in zebu dairy animals. So we believe that being able to combine stacking edits along with our heat tolerance really has a promise to transform the matrix of animal improvement production through by being able to adapt our animals to match the environment, so genetics by environment, and then also providing in places where veterinary intervention may be lacking, the ability of the smallholder to better manage their cattle. So we are really focused on tropical markets, and we see that this technology can have the biggest impact there. And we're talking about regions in low middle income countries where the amount, let's say on the dairy side, where there's a huge yield gap in the amount of milk that's produced per animal. And that's very clear where these blue circles are. You can see that production per animal is much, much lower. And part of that is because the starting genetic stock is either zebuine, which has not been improved as much on the dairy side as the Holstein and Jerseys and other European dairy breeds, but also native cattle have not been under selection very intensely for improved milk production. So there are some NGO programs going on to do that. but wouldn't it be great if we could bring in gene editing and really close that yield gap by bringing in animals that already have elite performance, but they're also adapted to the tropics. So our idea was to create the Thamani Holstein, and Thamani means, in Swahili, it means valuable or precious. So these are animals that we're breeding specifically for the African market. Of course, they could be used in other tropical markets, but the intention here is to really make them viable for the smallholder to use as a genetic alternative. So our concept was to select donor breeds that have proven genetic potential for milk, and we would need to add the regional adaptation. The native genetics, so our target edit selection, so what edits are we going to do, is really looking at native genetics for local disease tolerance. So we do that through population genomics to try and identify alleles that we can introduce that may provide better resilience to endemic diseases. And then, of course, heat tolerance is the lead trait. So we're stacking those traits together. The issue is that nobody had really done multiplex or changing multiple locations in the genome in a single transfection or in an embryo with great success. And part of that issue is fibroblast cells just don't last that long in culture because they're not immortalized. And you really need to be able to have cells that are durable, and we can do this HDR process so that we can end up being a non-GMO in a lot of the countries that have regulatory systems based on the Cartagena Protocol. And then, of course, we need to test those animals to prove they still have the performance and they're expressing those phenotypes from the traits we've introduced. And then providing the smallholder with breeding systems and extension information as to how to breed those in future generations. So part of our idea was to fund fellowships for 2 students from sub-Saharan Africa to help flesh out these issues. So we wrote a proposal back in 2020 during the COVID year to Bill and Melinda Gates, and it really had 3 prongs: a technical investment to move the technology along, so being able to create live animals that had multiple edits. The business side was to develop a pathway for regulatory approval and commercial acceptance to be able to bring these genetics to sub-Saharan Africa. So that's no small task. And then a societal impact would have been that these animals are durable because many of the smallholders use these as a bank account, and they can provide a sustainable revenue gains up to double of what they currently get from their dairy production systems. And of course, we had many partners that are helping in this project. The overarching goal was to produce 16 founder animals, 8 from each of 2 different breeds, and 4 of each sex within those breeds. The base breeds we're starting with were Gyr, because we know this is a breed that is somewhat improved and is used heavily in Brazil to help create the Girolando. The reasons being that it's already adapted to tropical conditions. It doesn't have the highest milk yield, but there are varieties within Brazil that do easily give 18 liters per day. So those are the top animals. And then, of course, we were going to use Holstein on the other side, the taurine side, and they had to be adapted for tropical climates. And we wanted to actually use Holsteins that had a little bit higher scores for their genomic predictions on functional traits and were more moderate in stature because we didn't want the smallholders to be having to feed or water them more than the regular genetics that they're using already. So really the goal at the end of this project is the smallholder has access to 2 different breeds that he can create his own variety of Girolando, which would mimic the 21 million cows that are in milk production today in Brazil that has made them near an export country for dairy. And really these animals average between 8 and 20 liters today, somewhat higher if you have that higher Gyr genetics in the background. And these, of course, would be more suited to tropical climates than regular Holsteins and potentially more sustainable because they're more resilient when there's lack of food, which happens sometimes in drought conditions. So what targets did we select for this multiplex genome editing? And here I'm showing one of the GIR animals that we made. That's Antonio on the side with his surrogate mother. He's a pretty handsome guy. We look at One Health. One of the diseases we wanted to address was infection by M. bovis, which causes bovine tuberculosis. And we know that raw milk consumption can cause transmission of bovine tuberculosis to humans, and up to 5% of the global population at any time has contracted bovine tuberculosis. So making animals that would be somewhat resilient to that and wouldn't transmit it, it would be good for the marketplace. We went through a number of genes with Dave McHugh's lab at the University College Dublin. He's an expert on working on bovine tuberculosis. And through their functional genomics data, we identified a number of hub genes, and then we compared the allele frequencies between Bos indicus and Bos taurus for those genes. Anecdotally, knowing that Bos indicus are resilient to tuberculosis, whereas Bos taurus aren't. You'll see a number of other publications out there in the literature looking at the same sort of contrast. The one that we focused on was this gene called MARCO, which is expressed in macrophages, which is the infected cell by M. bovis. And you can see that the alternative allele is fixed in most of the zebu breeds emanating out of the Indus River Valley region. And it's quite prevalent in admixed Sangha and other zebu breeds in Africa, and it's just non-existent in the taurine breeds. So that was a good candidate, we thought, for Holstein to bring tuberculosis resilience. On the GIR side, we know they don't milk quite as high, so through studies I was with at the University of Minnesota and looking at their milk selection genetic line, we found a signature of selection over chromosome 20 over growth hormone receptor, which matches previous QTL work done by Michelle George's lab. And so we thought introducing the Holstein allele or the taurine superior allele for milk yield would be a good addition to the GIR. And we genotyped many GIR that had the highest milking frequency in Brazil, more than 200 of those, and we never found this taurine allele in any of those GIR cattle. You could find the DGAT allele, which increases or decreases fat percentage in this case for the GIR. So we didn't want to really mess with the nutritional composition of milk, but increasing the milk yield itself we thought was a pretty good way to help improve those GIR. So this just shows the allele frequency data in Africa, and the allele is pretty much non-existent, even in the native breeds. Another disease we addressed with our edits was trypanotolerance. We know from QTL studies that were done at ILRI in the early 2000s, when they made a F2 population, they identified several regions, QTL regions within the genome, where that trypanotolerance was actually coming from the Indama breed, whereas the Boran breed would be susceptible. This disease is caused by tsetse flies. When they bite the cattle, they transmit a parasite, and that parasite causes nagana, which is a type of anemia that only the Indama are resilient to. Most European cattle succumb to that disease. So we identified 3 different loci that we were interested in. These were backed up by further population genomic studies that were done by my former colleague, Isu Kim. And in all those cases, we looked at the alternative alleles, and we could see that they were nearly fixed or high allele frequency in the West African cattle breeds, which overlaps with the region where trypanosomiasis is most prevalent. So we've identified our gene editing targets. It's very important that we genotype the founder animals' genetic lines we're going to look at, because you don't want to try and edit something when that allele already exists. You want to actually be making a change that's going to be effective. And once we had selected those donor animals, we decided on a process where we were going to actually clone embryos instead of animals. So we contracted high-value matings, and here I'm showing an example of 2 different Holsteins. Create those IVF embryos. The inner cell mass is turned into an embryonic stem cell line. that embryonic stem cell line can then be tested for pathogens. Sometimes you get BVD infection from media. So you want to make sure that your cultures are clean because it does affect cloning rate. We also can do genomic selection if we had multiple cell lines from a mating, and we could pick out those matings that had the highest estimates of genomic merit. And then those cells are ready for gene editing, so we can add our editing, and a process of bringing 4 edits. In our laboratory, we do it 2 at a time. We can complete this in 60 days. So this is just an example. We were able to clone the larger animal on the right there, where we only added the slick trait. And then in the second round of editing, we added 2 of the trypanosomiasis alleles, and that created the bull on the left, which is Alex. So those are 2 exact clones of each other. It's just that the one on the right has 1 edit and the one on the left has 3. And what we've shown is that this embryonic stem cell really has a high value in this process of making precision bred animals, because it has that longevity in culture, which does not affect cloning rate, and it allows us to deploy multiple edits. And the beauty about this, of course, is we don't have to worry about mosaicism. So all of our animals can be edited to homozygosity, and they'll transmit those alleles to all their offspring. So we were able to make 5 different Holstein sires in this project. The top 3 have already been collected for semen that's exportable. And these guys are now retired down in Texas because they're all slick. And we're still working on Ace. He's still very young, but he's in collection right now, going through his disease control diagnostics first. And Arthur will be being shipped off to the stud here in the next month or 2 as he passes a year of age. We also had 6 females. We've collected embryos from all of these girls, and we've bred most of them to our sires that were shown in the previous slide. Interestingly, one of the cell lines gave us 3 different animals, so the rate of cloning was actually quite good, and we were running at about 20% live animals from the number of transfers that we made on these multiplex gene edited clonal lines. On the GIR side, we were a little bit slowed by COVID and the fact that we tried to make these from a different type of cell, mesenchymal stem cell. We found that those were very hard to culture and did not work well in the multiplex system. So we had to access embryos in the United States, something that is very, very difficult when it comes to the GIR breed, because there's not a lot of elite genetics for GIR in the US. It's not a very popular breed. I know that a lot of elite genetics have been run from Brazil through Canada into the US. These 2 are actually from genetics at ST. And we were able to introduce 4 edits, 3 for trypanosomiasis and 1 for milk yield. And then we were able to get some fibroblasts from some of these high value 18 liter per day type genetics out of Brazil. And of course, with fibroblasts, we're much more limited, so we can only do 1 round of editing, but we added the milk yield in one of the trypanosome edits itself. And in one of those lines, we actually got 3 live calves. So that project's ongoing. We're very focused on clearing the path to be able to distribute those genetics to the smallholders. So that requires regulatory approval and a lot of work with researchers on the ground. So collaboration with colleagues in sub-Saharan Africa at the different agencies. But some of the work we continue to work on, of course, is non-GMO disease resilience. So these are different examples that I'll talk about. One has been gene editing in CD46. CD46 is a gene known to be involved in infection by BVD virus, so bovine viral diarrhea virus. The vaccines that are available for this are not very effective. And of course, this causes problems not only with the digestive tract, but also reproduction. And there's things known as persistently infected animals, which continue to spread this disease within certain dairy herds or beef herds. It's a big, big problem for cattle entering into the feedlot in the US. It's kind of the gateway pathogen that can induce bovine respiratory disease. And once you bring an animal in that catches BVD into a feedlot situation that was naive, and it's in one of these antibiotic-free type feedlots, you can't treat it. So being able to create some resilient animals could have a very good effect on improving animal health in the feedlot as well. So we made an animal with a 6 amino acid swap with researchers at Nebraska and also at US Mark. This animal just happened to be Jeer because we knew this cell line clone very well. And so we made that amino acid swap, and she was tested along with a naive Holstein animal. And of course, the result is that Ginger was very, very resilient. We didn't show any evidence of viral replication in her. So this trait still needs to be studied. And we have raised up Ginger now and bred her, and she's had a calf, which I'm sure there'll be news about that later this year. So it's proving that she functions normally, even though we made changes to a gene that clearly has more than one function. So we never planned on bringing this animal to birth. We were just trying to create cell lines from fetuses. So it's very important that you show when you're editing a gene that may have dual function that you still get good development. And that's what we've been able to prove in this rational design. So this is not any variation that already exists in nature. We're introducing this variation through changes in the code of CD46. Another example, of course, is PRRS-resistant pigs. There's been a lot of work done by the Roslin and the University of Missouri and Genus on removing domain 5, which is expressed in exon 7 of CD163. We found our own solution also in CD163, and we were able to obtain patent claims that are allowed for this. And what we're deleting is what's called the PST2 domain in exon 13. So just removing exon 13, we showed that this deletion also gives the same effect of making a PRRS-resistant pig. So we're actually in the second generation of testing these pigs. Of course, the issue here is that the time it's taken Genus to get to market and the amount of investment they've made has really probably dimmed the outlook of the other genetic companies for making PRRS-resistant pigs themselves to compete against Genus in the market. So that's just something to bear in mind. And part of that is just the burden of regulation in the United States as far as how long it takes and the fact that you have to compost all of those animals that are in a nucleus breeding herd until you're actually allowed to do commercialization because they're not allowed to enter into the food chain, even though I can't think of any risk that there would be from deleting a domain in a gene as far as causing some food safety issue. So in the end here, we know that using this technology, commercial deployment is possible, but it is very, very challenging, as has been witnessed through the press that's been put out by Genus and the seminars they give on bringing this into the swine industry, and also through this talk that I'm just giving you now. Some examples of those things that we have made to market, of course, are these slick Angus animals, where we did get commercial decisions in 2021 from Brazil as non-GMO. We also got a limited enforcement discretion from FDA in March 2022, and we're currently working to get a decision for the entire trait to be allowed into the market, which we hope will happen here in 2025. But it's a much more rigorous process to prove that it does the same thing in dairy animals, whether we're cloning or doing embryo injection. So we have to account for all of the quality control in those processes to show the safety. We'll say that a number of calves out of this Stay Cool bull have already hit the ground. So there's a yearling calves already in Hawaii. We have studies at the University of Florida. Calves have been hitting the ground there since November, and in mid-January, there's going to be calves coming to ground at Texas A&M. The great thing about using the semen from this bull is for every 4 calves he sires, 1 of them is normal, and 3 of them are slick. So what that means is you can have a large cohort of half-sieve animals, all derived from Stay Cool, and you can compare directly what the effect of slick versus non-slick is in these different environments. So we're really looking forward to those studies, and also one that's going to be done at the University of Missouri to test how slick does on fescue toxicity. We are still in the process of exporting to Brazil our products, and that includes the work that's going to be done by Professor Church there in Canada with his colleagues in Brazil. But again, we're just going to demonstrate that the slick has that same difference for environmental resilience to allow these animals to express their full genetic potential in tropical zones. So we know that right now, Angus in Brazil, especially north of Sao Paulo state, just don't work. We've also gotten commercial decisions for the first gene-edited slick Holstein, which was Diego. I showed in an earlier slide. So he's now been approved non-GMO in Brazil and Colombia. And this upcoming year, he'll obviously be allowed to market in the US if the slick trait is approved. And then we have dossiers already prepared to submit to Nigeria and Kenya, and we hope to get that done before the end of this year. And they will follow regulatory paths that are very similar to Brazil and Colombia. And then we're working on countries that still don't have their regulatory frameworks up and running yet. So an example of that would be Uganda, where we're collaborating with researchers from Iowa State and NARO in Uganda to test some of the slick genetics in the Ugandan smallholder environment. Of course, we have many other animals. These are going through approvals in the US and also in Brazil and Colombia. And the one on the right there is Durango, another slick Holstein bull. And there's a number of females that we have as well from the project we did with the University of Florida. And then next in line would be polled full-blood Wagyu. So this is genetics from Takeda, which was an allowed Japanese import into the United States of pure Wagyu, so full blood. So many of the US genetics are derived from these lines. However, these are going to be the first ones that are polled that exist in the world. And so we believe these genetics would be quite popular, especially if we can target their use in Australia. We're still working out differences in the regulatory system there, whereas this would be considered probably a non-GMO food by FSANZ, which is the food standards group that regulates the food. But on the genetic side, this animal is GMO because we have to use an oligo splint in order to create the polled allele, which is just a duplication of 200 and some bases in a region of non-genic region of the genome on chromosome 1. So all of this regulatory is done, all of our R&D is done, and we're able to make these commercial products. Can we still be able to sell these in the market? So just some commentary to wrap up here. I think, government policies just aren't keeping pace to support innovation and use of this technology. If it's too expensive to use and you have all of these requirements on traceability and labeling, differences in the trade barriers for export of the genetic products, and certainly there is a funding bias of the crop side gets much more funding than the animal side. We're usually decimal dust, and that's shown through the amount of already GMO or gene edited products that are in the market for animals. We just don't have any bearing on the revenue for the GDP in the United States at all. I think trade and breed association approvals also have to be done. So we have to do these outreach because these are the people that are going to be using these traits to do genetic improvement. And it's on a case-by-case basis. Is this a trait that they want to introduce into their breed? So we'll see here pretty soon, hopefully in January, as to whether or not we can create the first polled line 1 Herefords. That would be coming up. But we really need the trade and breed associations to be involved in the discussion at the table with the policymakers and the regulators as to how this technology needs to be regulated to make it functional. Of course, if you don't have shared benefits with the consumer, That's a critical part to acceptance, in our opinion. We know that the consumer is most focused on price and quality before they worry about GMO, except for those people that will never buy anything that is GE or GMO. And then there has to be some sort of standardization on trade, exchange, and labeling as well. Just an example of how far the activists will go to create fear in order to raise funds, because that's how they work. It's fear-based money raising. This is a pamphlet from Friends of the Earth that came out, I think, in 2016 or 2017, when the OGTR in Australia was going to deregulate SDN1, or just breaking a gene, and call it mutagenesis instead of genetic engineering. And so to counter that, they came out with this. And of course, you see these pictures of the Belgian blue, but if you open it up and start reading, you find out that these are actually Monsanto cattle that have been genetically engineered to have Arnold Schwarzenegger muscles. So complete fabrication of the truth, but people buy this and they give money. So in the US, the way that we're regulated, it's my belief that we do not get approval to go to market until we provide the FDA with all of the information they need to counteract activist litigation against those regulatory decisions. And I think Aqua Bounty is a perfect example of that case. So in summary, I'd just like to say, shared benefits from the technology, that's a big focus for us. And really, we've been honed in on doing adaptive traits from native cattle into performance breeds to make the marketplace more global. So creating emerging markets for genetic products that are going to have an impact on livelihoods, especially where the production per cow is very, very low. So this past year, we were funded through a collaboration with LIC that received another Bill and Melinda Gates Foundation grant. And we're going to create 20 different sires from their black and white grazing dairy based genetics to bring into the sub-Saharan African market. So that project's just kicking off right now. And so our take home is really true climate smart breeding includes the use of gene editing technology, because what better way to adapt your animals to the environment to bring in traits they don't have. And I think, our future is looking at direct measures to bring this technology for rapid genetic improvement, especially into politically challenging tropical environments. How do we keep it sustainable through the breeding programs? How do we make it available? And really, those are things that we're funding our students for, to help us provide leadership from their home countries as to how we might do that. So with that, I'll end and be happy to take any questions. Thank you for your attention.
Thank you, Dr. Sonstegard, for such a great presentation. I will now open it up to any questions in the room or online. And I think we already have a question in the room. So Bruno, go ahead.
Yes. First of all, congratulations for your talk, Dr. Ted. I am a big fan of gene editing technology, and for sure, the future of the agribusiness pass through the gene editing. I have a question for you about how to avoid the excessive use of a single animal as a carrier of a desirable allele, or how to avoid the negative effect of inbreeding and reduced effective size. I mean, you have one animal carrier of a desired allele. How could we avoid the overuse of this animal? Have you considered, for example, first spread the desired allele through a F1 generation or F2 generation, and then use those animals as breeders to spread the desirable allele through the herd?
Yeah, so I think it's a great question. And this is, we're not like plant breeders where we just need one seed to multiply out. We need a lot of diversity in our animals. So you have to create many, many founders. And I think that's very evident from the amount of funding that had to be invested by Genus in creating their nucleus breeding herds. They couldn't just do it from a single pig edit. They have to bring it throughout the population. So you need to have multiple homozygous animals of different genetic varieties, and you can model all this out. I mean, we've modeled it out for what would it take to make a line of completely heat tolerant Angus without running into those inbreeding problems. So it's just a matter of creating a number of founder animals and then bringing in new genetics every year. So that's good for our business, because if we get paid to make founder animals, of course, that's the best outcome for everybody. You remove that inbreeding, and we continue to be able to use our gene editing technology to introduce these traits into new genetic lines to expand that diversity. I think the idea of just making one animal in a commercial market, yeah, it's a great novelty, and it may generate some income, but for it to be sustainable, you have to have multiple customers or multiple animals within a genetic breeding program in order to do it.
Okay, thank you.
Thank you. Colin, did you want to go ahead?
Yes, first, thank you, Tad, for the great talk. My question kind of comes and relates to the diseases that you're kind of potentially going to be looking into, and specifically around the phenotyping of those individuals, because you want to look at one specific virus that is causing the disease. And I wonder with things like diarrhea and respiratory disease, where you can have a whole host of different things potentially causing it, how do you ensure, for example, that your environment is controlled enough that the animals are only exposed to one? Is it specific, like really closed group of animals? And then a question on that would be, do you have enough genetic diversity within that closed group of animals to actually represent the population, and kind of, or just what your thoughts are kind of around that?
Yeah. So this is not our company's expertise. I would guess our part of the expertise in that process would be to find collaborators that have the facilities and the means to do what you're talking about. So having a sterile environment until the time of challenge, having expertise and understanding the epidemiology of the disease. So when we did the work on ginger, it was very, very difficult because we had to keep them in a closed room for a long time, isolated from getting contaminated. We had to watch out what kind of milk replacer we use, because you could contaminate them or give them passive antibodies through that. So there was a lot of background work that needed to be done, and we had to wait a long time for those animals to clear those antibodies out before we could actually do the challenge. So that's not my expertise, but that's why we need those collaborations. You know, if we're looking at some of the other diseases like trypanosomiasis, I'm going to have to use some of those elite genetics we've created to validate those phenotypes in the African environment. There's very few places that do that research here in the US. There's maybe one. So I would just prefer to do those on the ground there and provide those research institutes there with the ability to evaluate those, because coming from them is going to be much more valuable for the smallholder than coming from some multinational company as far as what the results are. On the TB side, we're using in vitro challenge. So we actually, the experiment that one of the students is going to do is she's going to draw isolate monocytes out of the blood from her edited animals and age-matched non-edited controls, and then take those through a challenge experiment, which those protocols are very well documented. And so we're going to look at a molecular phenotype to see, how the gene expression changes and whether or not those are indicators of resilience. So we also have some good positive controls because we have zebu animals that we can compare to as far as what the molecular response to expression is going to be from those animals that are resilient to the TB. So those are just some examples.
No, for sure.
It takes a long time, and if you're going to make claims with the regulators that you have disease resilience, you have to have the data to prove it or they won't allow the claims.
Yeah, for sure.
And it's expensive, right? So.
Perfect. Thank you, Tad.
Yep.
And okay, there's one question in the chat that asks, how is it possible that genetically modified animals are labeled as non-GMO? What has to happen to get a GMO animal?
Yeah, so that's a great question. So according to signatories of the Cartagena Protocol, and I'm not an expert on this, but they define a living modified organism within that diplomatic charter. And so Brazil and Argentina, for example, use that diplomatic charter to form their regulatory framework. So if you introduce foreign DNA, like a gene from another species, or even a gene from the same species, but put it in a place where it's normally not located in the genome, that is going to be considered GMO according to their definition of what GMO is. If you are just breaking DNA and recoding it by changing a few bases, or maybe even through the polled animal, just, changing a non-genic region, um, their ability, because of that Cartagena protocol, is to define it as non-GMO. That means it gets treated under conventional regulation. In fact, the regulatory process in Brazil, for me, it's much harder to import any genetics at all into that country uh, compared to going through the regulatory system that they have, which is volunteer scientists who are very knowledgeable and capable to identify whether something is GMO or non-GMO. So from those countries in Brazil and Argentina, they've gone out and done helped countries in sub-Saharan Africa and Pac Asia develop similar systems. So there's a whole group of countries now that are very well aligned as to how they'll evaluate gene-edited animals, whether or not they're GMO or not. Australia, India, the EU, Canada, the US, and Australia. I already said Australia. They have very different regulatory systems that are not based on this GMO, non-GMO definition per se. Australia is some, but theirs is a different definition than the Cartagena Protocol. So I hope that answered your question. It's very complicated.
We do have one last question if you have time, Dr. Sonstegard.
No problem, no problem.
Perfect. Carolyn, if you want to go.
Yeah, thanks a lot. Thanks a lot for your presentation. It's really interesting. I was just had a question about the SLIC mutation because I've heard a lot about it from Dr. Church. So what is your experience with this SLIC mutation in colder environments? Like, for example, do cattle suffer increased cold stress? Because just if they didn't, that SLIC mutation might become more mainstream throughout the breeds that start to have those types of cattle.
Right. So before gene editing was on the scene, Senapol was a thing where they were importing that from St. Croix into the mainland US. And I know there was a number of beef breeders that were using Senapol bulls, creating some purebred lines. They would take those calves, crossbred Senapol, but they were probably slick, and they would send them up to feedlots in Colorado. And I don't know if it was the Senapol themselves or the slick. They didn't perform very well. So, that ranch, for example, that I'm talking about, dumped breeding Senapol. Part of that is just the original stock is not very good. We don't have a lot of data points, but those two animals I showed that have been approved, those have both gone through minus 20 degrees in Iowa. And Stay-Cool was giving semen there. He also was in Colorado producing semen. We never had any problem with him. He grew some hair. He didn't grow a lot, but as long as he had dry bedding and a place to get out of the wind, they were fine. Long term, I think you have to do a much larger study. I know there's Angus breeders that do breeding in the North Central Plains, and they send a lot of their bulls down to Florida. They're very interested in knowing if there's any effect on those animals with the cold. So it involves more research. I know that's something that John's been working on in the past. So we'd be happy to learn that, because a lot of guys that breed cattle in the South are going to send their animals to northern feedlots, or at least the middle America, and they're going to be concerned, am I going to get docked because my animal's slick, even though it helped me get my calf to weaning at a heavier weight. So
Yeah, thanks.
Good question.
Could I just make a quick comment?
Sure. How you doing, John?
So, I'm good. Thanks for the call out. So I have quite a few St. Paul Angus and calves up here in Canada, and there's no question they grow. We've crossed them with cold tolerant breeds like Galloway and Scottish Highland. The slick gene is compatible. Actually exclusive. They seem to be able to coexist. That was Graham Plast. So likely not. I think in the future, both. We could have a that could be adapted for both climatic extremes, and I think that's critical in North America. Here in British Columbia, we've got whole industries that have already been wiped out by climate change. Our vineyards are gone, our orchards are gone, and I'm terrified at the prospect in the future that the way climate change is going, we start getting summers like we did in 2021, where the temperature goes over 50 Celsius. We're not going to be raising cattle outside very long if that continues. Thanks.
Thanks, John.
All right, I think we'll cut it there. Thank you for a great discussion, and thank you again, Dr. Sonstegard, for presenting for us today. The recording will be posted to YouTube, and we can send you those details later. Thank you again, everyone, for coming and for such a great semester. Enjoy the holidays, and we will see you in the new year with our first seminar on January 17th.
Thank you. Happy holidays.